So we were talking about the classes of antibodies or classes of immunoglobulins, and we have already discussed that primarily there are 5 classes of immunoglobulins. One way to remember is GAMED, GAMED. GAMED stand for what? Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. Already we have discussed in detail about immunoglobulin M, right? Now we will talk about immunoglobulin G. Why immunoglobulin G is called immunoglobulin G? Because the heavy chains in the immunoglobulin G belong to the gamma group. Is that right? Now, what are the important points related with immunoglobulin G? Immunoglobulin G is number 1. It is the most abundant immunoglobulin in your body. Most abundant immunoglobulin. You know why? Because with repeated infections, immunoglobulin G keep on accumulating, right? You must be knowing that immunoglobulin G is the antibody of secondary response. Again, as we mentioned previously, that let's suppose this is the time, here is immunoglobulin concentration. If there is repeated exposure, this is suppose first exposure, second exposure, third exposure to the same antigen. So initially the immune response which will come, that is called primary immune response. After that there is secondary immune response, and with repeated exposure there is now another secondary immune response. Again I will repeat, as I mentioned previously, there is a big difference in primary immune response and secondary immune responses, right? Why? Number 1, in the primary immune response there is a slow and gradual increase in antibody concentration, and it reaches to the peak at low level and gradually it comes down to very low level. And the main antibody in the primary immune response is IgM. This is mainly IgM, right? In primary immune response, concentration of IgG is very low, right? But with every new exposure, suppose this is our second exposure. Now, when the antigen comes second time in your body, it will find some of the memory cell, right, which were activated in the previous response. And those memory cells will rapidly proliferate and give very strong antibody formation response. And now the antibody will be dominantly IgG, right? But during this time, right, after the first exposure, for example, after some time when the second exposure will occur, during this time from the bone marrow, some new B cells may have come and they will be taking the second exposure as their first. You are getting it? And they may produce what? Another IgM production. So what really happens with repeated exposure? With repeated exposure, IgM is produced with every exposure, right? But in the primary exposure, on the first exposure, IgM is the main antibody. As far as IgG is concerned, IgG is during the first exposure made in very small amount, but with repeated exposure, concentration of IgG keep on increasing. Why the concentration of IgG keep on increasing? Because the cells which originally made IgM, now they have switched, they have done the class switching and they have started making IgG. When there is repeated exposures, it means during every exposure, some plasma cells are, B cells and plasma cells are made, and they initially make IgM and then switch to IgG. So when you have repeated exposure to the antigen, you have lot of B cells, memory B cells, which are ready to produce lot of IgG. Is that right? Another thing which is very important to remember, half-life of IgM is short, but half-life of IgG is long. That is why if you get antigen today, you will make IgM, right? But gradually IgM will its half-life is less, so gradually its levels will go, rather not gradually, rapidly in your blood, the IgM level go down. But in repeated exposure, when you make IgG, because its half-life is longer, it will stay in your body longer time. Am I right? Now, let me mention one thing again, that what really happens, that if I get infection today, first time, then against that antigen, I will be making IgM, right? But if I got some infection 5 years back and again I get the same infection today, then I will make IgG because I have a previous exposure, and previously you can say previously trained memory cell will be reactivated and they will produce IgG, and some new B cell will also be recruited and they will produce IgM. So what really happens, it's a very important concept, that if against an antigen you have high titers of IgM in your blood, it means that infection is recent. But against another infection, if you are having only mainly IgG in your blood, then it means this is the past infection. But if you have right now in your blood against a microbe, IgG as well as IgM, it means that you had previous exposure to that organism as well as you have current exposure. Am I really clear? Right? So, what I am talking about, IgG is the most abundant antibody present in your body. Number 2, it is the antibody mainly in the secondary response, right? Thirdly, IgG is present as monomer. It is monomeric. Again, keep on comparing monomer. Keep on comparing with the IgM. Look, IgM was the third most abundant in the blood. The first most abundant is IgG, then second most abundant is IgA, and third most abundant is IgM. Again, the concentration of antibody in the blood decreases according to the sequence of alphabets in GAMT. That IgG has the highest concentration of the antibody, Ig has less than that, IgM is more or less, Ig is very less, and IgD is in traces. You get it? So, keep on comparing when we are studying IgG with the IgM. That IgG is the most abundant, IgM in the blood is less. Number 2, IgG is antibody of secondary response, but IgM is antibody of, yes, primary response. Is that right? Then IgG is as monomer, but IgM is present in the blood as pentamer, right? IgG circulate in the blood as monomer, so we can say that it is having 2 heavy chains and 2, yes, light chains, and heavy chains belong to gamma and gamma. Is that right? Because it is monomer and it's a small molecule, so not only it circulates in the blood, this also comes into other body fluids, right? So IgM is mainly in the blood, IgM is mainly in the blood, IgG is in all body fluids, but IgA, which we'll study later, it is specially secreted in our body secretions. Is that right? IgG is not present in our body secretions in much amount, right? Again, it's because it is monomer, so it must be divalent. It must be divalent. Is that right? Now, another thing which is important about it is that it crosses placenta. It crosses placenta. So it means that on the placental cells, you know, placental cells express the receptors for Fc portion of IgG. Let's suppose here is placenta and here is your fetus, right? Now, what really happens that the cells on the placental surface, the cells which are facing to the maternal blood, they express the receptors for Fc portion of IgG, and so IgG from the maternal side, from mother blood, IgG bind with these receptors, and once these receptors are loaded with IgG, they undergo endocytosis process, and then and through the pinocytosis and endocytosis, they are transferred from maternal blood side to the fetal blood side. So mother is giving a very precious gift to the baby. What is the gift? IgG. So IgG basically is a special maternal gift to the baby. So when baby will come to the world, he will have lot of IgG from his mother. Why IgM is not given to the baby and IgG is given? Is there any special advantage in giving IgG and not IgM?
IgM is too big.
No. Mother gives very big things to the baby. Now look, it's very simple. Let's suppose there is a Japanese mother who has come to visit America and she is living in America for few months. If she gives only IgM to her baby, she will give only those protection against those infections which she currently suffer. You are getting it? But if she gives her baby IgG, then in her blood she has IgG against almost all infections she suffered in her life. And usually baby live in the same environment in which mother lives. Fathers are a bit different thing, right? So what really happens that mothers, this is the best gift they give to their babies, that mothers have the pool of IgG, or every person has a pool of IgG antibodies directed against many microbes for which we have been exposed in our past years. You are getting it?
Yeah.
So if a mother is giving IgG to her fetus, so she is giving immunological protection to the fetus against many infections, not only against the current infection but also against her past infections. But if mother could give only IgM, then mother will protect the baby only against the current infection. Is that right? So it is a superior strategy which has evolved during evolution, that human females give to their baby transplacentally their own IgG, the pool of IgG. If a woman is living in Africa, right, she will have different pool of IgG. A woman who is living in USA, she will be exposed to different infections in her life. She will have a different pool of IgG. And woman who is living in India, she has been exposed to different set of infections, and she will have different types of IgG in her blood. So all these three mothers will give their fetuses different type of pools of IgG, which are best suited for the baby protection once they take birth. Am I clear? So there's a logic in that. Am I right? Okay, there's a very important concept. If a baby is born and baby is having IgG against the syphilis, newborn baby has IgG antibodies against the syphilis, but baby does not have any IgM against the syphilis. Do you think baby has been exposed to syphilis, Treponema pallidum or not? Let me repeat a question. A newborn baby has IgG against the Treponema pallidum, the causative organism of syphilis, but that newborn baby does not have IgM against the syphilis. What do you think, baby has been exposed to syphilis organism or not? No. Probably mother was exposed to the syphilis somewhere in the past and she had anti-treponemal IgG and she passed it to the baby. But if during the pregnancy mother is suffering from syphilis during pregnancy and Treponema pallidum through transplacental route, Treponema pallidum from maternal blood goes to the baby and fetus, and baby gets the organism of syphilis, such baby, if he takes birth, right, in such baby's blood you will find IgM directed against Treponema pallidum. Am I clear? So a newborn baby, if newborn baby has preformed IgM against an organism, it means that baby was exposed to that organism during intrauterine life. Am I clear? But if a baby has IgG against multiple organisms, then it means probably these IgG, not surely these are IgG molecules are coming from maternal side. So IgG is most abundant. It is antibody of secondary response. It exists in monomer, so it is divalent and it crosses placenta very efficiently. And how it works? Number one, it can activate complement. Activate complement system. Complement system by classical pathway. Classical pathway. Let me tell you what exactly is meant by this. Let's suppose here is a bacteria. Okay, let's suppose here is a bacteria and these are bacterial antigens and you are having IgG antibody, right? And as soon as IgG antibody bind with the antigen, its Fc portion become active and now it can activate complement. So IgG can activate complement number 1, C1, 2, 4, 3, C3A will go away, C3B will be there, then C5B, C5A will be away, and then C6, yes, 7, 8 and 9, right? This is membrane attack complex, this much, right? So basically IgG antibody, once it binds with the antigens, then its Fc portion is activated, which can activate the complement by classical pathway and kill the microbe. This is one way. Secondly, IgG can act as opsonin. Now what is meant by opsonization and opsonin? Actually, when IgG bind with the bacterial antigens, its Fc portion become active and neutrophils and macrophages are having membrane receptors for the active Fc portion of IgG. So if this is a neutrophil, then neutrophil will have receptor for the active portion of right. This is a neutrophil and neutrophil had this specific receptor and this receptor could bind with the active portion of IgG. So IgG on one side is holding the bacteria. Of course, bacteria is very sad, right? IgG is holding on one side bacteria, another side it bind with the neutrophil. Now bacteria cannot escape, right? And these receptors give signals into phagocyte, and these signals stimulate the phagocytic process. So actually, when IgG bind on the particulate matter, for example bacteria, it enhances the phagocytosis of these particulate matter of bacteria, right? This process is called opsonization. What is opsonization? Opsonization is a process in which opsonin molecule bind to a particulate matter. On one side they bind to the particulate matter, which may be bacteria or a cell. On other side they bind with the phagocytic cell and they facilitate the process of phagocytosis, right? So IgG act as opsonin. So IgG can kill the bacteria by activation of complement, or IgG can help the neutrophil and macrophage to very effectively phagocytose the bacteria which are opsonized with the IgG molecule, plus IgG molecule can neutralize, yes, what? Neutralize virus and toxins. Now, what is the concept of neutralization of virus? Yes, Dr. Safa, you will tell me something. Have you heard of this term? There are some antibodies neutralize the viruses or toxins. Okay, let me explain. Let's suppose this is the target cell, right? And this is a specific virus. Suppose this is a virus and this virus is going to attach with this cell and push its genetic material in. Actually, on the surface of the virus, there are special type of proteins, and these proteins help the virus to bind with the target cell, right? On the target cell, there must be receptor proteins, and let's suppose this protein of the virus will bind with this cellular protein. So virus will bind here like this, right? and when these two proteins interact, then virus will be taken in, or virus genetic material will be going in. So first step for virus to attack our cell is that virus should develop attachment with our cell. Once virus is attached, then it will push its genetic material in. Is that right? IgG antibodies actually bind on these proteins and block them. If IgG antibodies bind with these proteins, do you think now virus can bind here? No. So we say capability of virus to bind and invade our cell has been neutralized by the presence of IgG. This is the concept of that some antibodies can neutralize virus. Of course, IgG is one of the very important. In the same way, there are some toxins, right? Let's suppose there's a toxin which has A component and it has one more component which is called B component. Many toxins which are produced by bacteria, they have A component, A for action, which produces action in the cells, toxic action in the cell, and B is for binding for the cell. So let's suppose this is the receptor for the toxin. This is receptor for the toxin, and actually B component binds here, and only then when B component of the toxin bind with the receptor, only then A component will be translocated into cell, and then A component will do toxicity in the cell. Actually, some of the IgG antibodies, they bind with the B component, and when they bind with the B component, can B component bind with the receptor on the target cell, and can toxin enter into cell? So capacity of toxin in the presence of IgG against its B component, right, then capacity of the toxin to enter into cell has been neutralized. So this is how we say that this is a neutralizing antibody, because it can neutralize virus and toxins. Am I clear? Again, you must compare these functions with the IgM antibody. IgM could also activate the complement. So complement can be activated in a classical pathway by IgG as well as by IgM, right? Opsonin function of direct opsonization is done by IgG, but direct opsonization is not done by IgM, because there are no receptors on the phagocytic cell to bind with the Fc portion of IgM. So IgM cannot directly opsonize the bacteria, but IgG can directly opsonize the bacteria. Then neutralization of virus and toxins. IgG and IgM both can do this function. Another thing which is there, that IgM could do what? Agglutination. You remember IgM was as pentamer with decavalent, it could bind with multiple particulate matters. So IgM could work as agglutinating antibody. But IgG, because it is only divalent, can it cross-link many cells or particulate matter? No. So IgG cannot do agglutination reactions. IgM can do, but IgG cannot do agglutination reaction. But IgG can be involved in precipitation reaction. Precipitation reaction means that in a test tube you have put the antigens. There are many antigens, and then you add to this IgG. IgG and antigens may make, what is this? They may make antigen antibody complexes, and because these are small complexes, so they may settle into bottle, right? So they play a role in precipitation reaction. So IgG can play a role in precipitation reaction, but IgG cannot play a role in agglutination reaction. IgG cannot play a role in agglutination reactions. Agglutination means that antibody should cross-link multiple particulate matter, right? Like multiple bacteria are cross-linked and clumped, or multiple RBCs are clumped. This is a function of pentamers of IgM. Is that right? So these are a few words about IgG. Now we will talk about IgA. Now IgA, do you think IgA antibody, it is the second most abundant antibody in your blood. But the very special thing about IgA is that not only it is present in your blood, but in large quantities, IgA is also present on all mucosal lining of the body and in all body secretions, right? So that makes IgA very important antibody. Let me explain. Let's suppose that this is GIT and this is a GIT mucosal cell, and here is your blood circulation, and let's suppose here you have a plasma cell. Is that right? These plasma cells are producing IgA, right? Now, when IgA is released, now let's suppose these are IgA antibodies. Some of the IgA which are released, they are held together by J chain. They are held together by J chain. So it means they are released as dimeric form. But some of the IgA is released as monomers. These monomers will be going to the blood. So you must remember IgA circulate in the plasma in a monomeric form. IgA circulate in the plasma as monomeric form, or this IgA is also called IgA1, right? But they are not cross-linked by the J chain. But this these two molecules of monomers of IgA which are held together by the joining chain, and this is now becoming a dimer. This dimer is internalized by, it enters into, what is this cell? Mucosal epithelial cell, right? And once IgA dimers are inside, already they were joining with which chain? J chain. Then epithelial cells of mucosa will produce another protein which is called secretory component, and secretory component also bind with that. Secretory component. So secretory component is a small protein which is synthesized by mucosal cell, and this binds with the IgA dimers, and then secretory component help it to the whole complex to go to the lumen. So eventually this dimer of IgA, right, with the two joining and one, what is this? Secretory component. Whole this complex is secreted on the mucosal lining, right? These IgA are very good molecules because whenever virus or bacteria they are coming, those viruses will bind here, and then of course they cannot invade the mucosa. So many antigens which are coming to the mucosal lining, those antigens or virus or bacteria, they will bind with the pre-formed and pre-secreted IgA, and in this way they will be neutralized and they will not be able to invade in our cells. Is that right? That is why IgA is said to be antiseptic paint of our mucosal linings. This is also called that IgA molecules, dimers are antiseptic paint on our mucosal linings, right? It means, let's put it there, the important information, IgA is the second most abundant antibody in our body, right? Secondly, it is present as monomer in our, yes, it is present as monomer in our blood. It is present as dimers in, yes, dimers in mucosal surfaces plus body secretions. Number 4, that IgA, right, this is present in which mucosal membrane? Every mucosal membrane. Throughout the gastrointestinal system, it is painted with IgA. Urogenital system, it is painted with IgA. Your respiratory system, all respiratory mucosa is painted with IgA. Even your conjunctival mucosa is painted with IgA. So it's a mucosal defense mechanism. against the invasion of many microbes, right? Secondly, which secretions? All the body secretions, all respiratory secretions, gastrointestinal secretion, urogenital secretions, even in the tears, in the saliva, in the milk, and especially colostrum has a large amount of IgA. Actually, this is very interesting and very important to know that when newborn baby is born, baby is having a lot of IgG molecules transferred from the placenta to protect the baby against many infections. But after the birth, if mother is breastfeeding, then baby is getting a lot of IgA from maternal milk, and this IgA will line the baby's GIT mucosa so that if some bacteria or virus come to the baby's gastrointestinal system, they will be neutralized. Is that right? But mothers who are not doing breastfeeding, their children have more often gastrointestinal infections and diarrhea, because the formula milk which are coming, they are not bringing IgA in that. Is that right? So it is in high concentration applied on the mucosal lining. It's in high concentration present in the all body secretions including milk and colostrum. And then another important thing about IgA is that it does not activate the complement by classical pathway, but it can activate the complement by alternative pathway. So classical pathway, complement activation, no. IgG or IgM could activate the classical pathway, but IgA does not activate the complement by classical pathway, but it does activate the complement by alternative pathway. It can activate the complements by alternative pathway. After this IgA, now we talk about IgD and IgE. So let's talk about IgE. IgE is the class of immunoglobulins which are having heavy chains belonging to the epsilon group, IgE. IgE antibodies produced in small amounts as compared to the IgG, IgA and IgM. Right? IgE is in the blood in very small amount. Both of them are in traces in the blood. Now IgE, once it is formed, it loves to bind with the mast cells and basophils. IgE is also called cytophilic antibody. Cytophilic antibody means it loves the cell, right? For example, once IgE is made, suppose this is IgE. Again, it has two heavy chains and two light chains held together by disulfide bond. So IgE exists as monomer with divalence. But most of the IgE right now which is present in your body, that is bound with the mast cells, because mast cells have receptors for Fc portion of IgE. But when antigen bind here, only then this Fc portion become active, stimulate the mast cells, and when mast cell is stimulated, you know, when an allergic substance, actually when we say that, for example, if you have allergy to penicillin and you have made anti-penicillin IgE antibodies, then IgE antibodies will be loaded on the mast cells, and when allergen come bind with the IgE antibodies, actually allergen usually cross-link multiple IgE. These are the IgE antibodies which are previously formed and loaded on the mast cell receptors. When the allergen comes, suppose this is an allergen, right, it will activate, bind with the multiple IgE molecules on the mast cells, and they will come together and then they will give signals to the mast cell, and mast cell will be activated. Number one, signals will go to the membrane, and from the membrane there will be release of prostaglandins and there will be release of leukotrienes, plus there will be release of lipoxins, right, plus there will be release from the membrane of platelet activating factor. So what really happens that when IgE is activated on the surface of mast cells or basophil, Basophils are circulating into blood. Mast cells are the connective tissue cells present in your connective tissue throughout except central nervous system. Central nervous system does not have mast cells. So when IgE is loaded on the mast cells, it activates number one the membranes, and from the membrane lot of chemical mediators of inflammation are released like prostaglandins, leukotriene, lipoxins and platelet activating factor. Plus they give signal to the cytoplasm, and these signals when they come to the cytoplasm, number one to the membrane, number two signals are going to cytoplasm, they activate the granules and mast cell degranulation releases, yes, histamine. Do you know that? Histamine, 5-hydroxytryptamine, plus it releases proteases, proteolytic enzymes, plus it releases, yes, neutrophil chemotactic factor and eosinophil chemotactic factor. So what really happens? Once allergic substance bind with the IgE, which is already loaded on the mast cells, mast cell membrane activation releases lot of inflammatory mediators, as well as mast cell cytosolic granules release also produces chemical mediators which enhance the inflammation, and if even signal will go to the nucleus, and nucleus will lead to formation of new messenger RNA, which will translate into proteins, and some proteins will be secreted out which are actually cytokines, like it will maybe releasing tumor necrotic factor or interleukin 1. So IgE plays a very important role in activation of the mast cells and basophils during type 1 hypersensitivity reactions, during which all these chemical mediators are released into tissue and produce severe inflammatory reaction. Am I clear? Then IgE not only binds with the mast cells and basophils, but IgE plays a very important role in defense against parasites, especially helminths. For example, against Scarus, strongyloids, or against hookworms, against trichinella. These are different type of parasites. People who are infested with parasites, they have very high level of IgE. IgE, which is directed against the parasite antigens that help us to kill the parasites. For example, if there is a parasite here, right? Of course, these are the antigens of the parasites. IgE will bind on the parasite antigens, and IgE also has its receptors on the what are these cells? This is not mast cell, this is eosinophils. These are eosinophils. So what really happens that IgE antibodies can coat the what are these parasites, and their Fc portion can activate the eosinophils, and then eosinophils will release very important destructive products, and these destructive products have one important function which is called major, yes, basic protein. And this major basic protein and some other substances which are released by the granules of the eosinophil, they destroy the parasites, right? In this way, IgE helps us to kill our parasites, right? And the people who are having these parasites in their body or the larvae of these parasites, these people have high concentration of IgE. And now we come to the last antibody group, and last class is IgD. The good news about that is we don't know much about that. This is present in the blood in very small amount in traces, and it is present on the B cells as B cell receptor. You remember B lymphocytes were having on their surface IgM, but there are also some IgD. So IgM and IgD are present in monomeric form, expressed on the surface of the B lymphocytes, and they act as receptors for the antigens. So B cell receptor or BCRs are mainly IgM and some of them are IgD. Is that right? So, okay, I will ask you some questions. Now we will talk about another way to type the antibodies. Antibodies can be classified as idiotypes, allotypes and isotypes. Another way to classify antibody is allotypes, isotypes and idiotypes. Now listen, let's suppose here you are. And from your circulatory system, we remove all the antibodies. We remove all the immunoglobulins. Now all the antibodies which we can bring out from one person, they have something common. Right? Because antibodies are what? Antibodies are just peptide chains. These are 4 peptide chains, 2 heavy chains and 2 light chains held together by disulfide bond. Is that right? And antibodies, because they are protein, so they are antigenic. For example, if your antibodies are injected into a donkey, suppose this is a donkey. I don't know how to draw a donkey, but you can imagine it. This is a donkey, right? And if human antibodies, suppose antibodies from this person are injected into donkey, these are the antibodies. When antibodies will come, do you think for donkey these antibodies are self proteins or foreign protein? It depends on the person actually. Right, human antibodies for the donkey are foreign proteins. So donkey's immune system will take these human antibodies as antigens, and donkey's immune system will produce antibodies against human antibodies. You are getting it? So it means human antibodies are antigen because they are proteins, they have antigenic areas, or we call them, they have antigenic determinants. Is that right? Now, actually, they found that all the antibodies which are present, removed from one person, all the antibodies which are present in one person, they are having something common. For example, some amino acid sequence here in the light chain and some amino acid sequence, let's suppose here. This amino acid sequence is present in all the antibodies of this person, right? So, according to this concept that this person has inherited a special type of alleles, and those special inherited alleles, all the antibodies from this person have some amino acid sequences which are common, right? And according to these amino acid sequences, antibodies vary from person to person. For example, in my case, in my all antibodies, suppose here is histidine and here is leucine. Maybe in your case, at this point, there is histidine and threonine. So my all antibodies have histidine and leucine, and your all antibodies have, suppose, histidine and threonine. So it means all my antibodies have same antigenic determinant here, and all your antibodies have a different antigenic determinant here. But for your antibodies, all of them will have the same antigenic determinant. So this, if we classify human antibodies according to this antigenic determinant, we say we are doing allotyping. So I have one allotype and you have one allotype. So all the antibodies which are present in one person, that is determining its one allotype. Is that right? Opposite to that, isotype means the differences in antibodies in the same person due to heavy chains. You know the constant of the heavy chain. These are the constant region of heavy chain. How many different types of heavy chain one person is making mainly? 5 types. Because these are the 5 basic classes. So we can say if we bring all his antibodies out and try to classify his antibody according to constant of the heavy chains, how many will be main group? 5. So if donkey is going to make antibodies, against the constants of the heavy chains, how many types of antibodies you will make? 5 types. Is that right? So this, when you classify antibodies in a person according to the constants of the heavy chain, they are divided into 5 groups, and those groups are called isotypes. What are they called? Isotypes. So I have how many types of isotypes? 5. And you have how many types of isotype? 5. And my isotype and your isotypes are same. This is very important. You have IgG, IgM, IgD, IgE, IgA. I have also same groups. Is that right? But donkey does not have the same group. For donkey will make anti-isotype antibodies. When you put my antibodies into a donkey, donkey will make how many types of anti-isotype antibodies? 5. But donkey will make how many types of anti-allotype? 1. I have only 1 allotype. So if you inject my antibodies into a donkey, the donkey will make how many anti-allotypes? 1. Donkey will make how many anti-isotype? 5. So one person has how many isotypes? 5. Or 1% antibodies, when they are classified according to allotyping, in one person how many allotypes are present? 1. If one person's all antibodies are classified according to the isotypes, how many groups are there? 5. Thank God you know it. Now idiotype. Ideo means unique. Ideo means unique. Actually, if I have been exposed to thousands and thousands of many different virus and bacteria, then it means I've made thousands and thousands of different type of antibodies with specificity for different antigens. Is that right? And why now one antigen is, let's suppose one antibody, you know, every antibody has different, there's a special variable areas here, variable area of the heavy chain and variable area of the light chain, so that this antibody molecule must bind with a specific antigen. Now tell me one thing. In one person, if you try to classify all my antibodies according to these variable regions, how many antibodies groups I will have? Millions, right? This is say that all the antibodies which are derived from, which are made by one clone of B cells, they have the same variable region. But actually, every person has capacity to make millions of different antibodies directed against different antigens. So it means in every group of every antibody, these variable regions are so unique that every antibody will bind with a unique antigen. Or to be more clear, all the antibodies which are derived from one clone of B cell, they are directed against a unique antigen, and all those antibodies will have a unique amino acid sequence here. Is that right? Now, if you remove my all antibodies and inject into donkey, and if donkey is going to make antibodies against variable regions of my antibodies, he will make millions of different type of antibodies. We say that he is making anti-idiotypes. Idiotypes. So what is idiotyping? Idiotyping is the typing of the antibodies according to their variable regions. Isotyping is the typing of the antibodies according to the constant of the heavy chain. Allotype is typing of the antibodies from person to person, right? So because every antibody which is directed against different antigen has a different type of idiotype. So what are idiotype? Idiotypes are the antigenic determinants made by the amino acid sequence in variable region. Let me repeat it, it's worth repeating. What is idiotype? Idiotype is the antigenic determinant present made by the amino acid sequence present in variable part of the antibodies. And one person may have millions of idiotypes. Then isotype. What is isotype? Isotypes are antigenic determinant in the antibodies of a person which depends on the amino acid sequence in constant of the heavy chain. Is that right? And allotype is the antigenic determinant present in a person's antibody according to the allelic morphology, right? Again amino acids. So I can say that all my antibodies will have one point unique, which is unique to my all antibodies, right? So as far as this is allotype, so how many allotypes I must have? 1. How many isotypes I must have? 5. How many idiotypes I must have? Millions. Is that right? So when we say that there is anti-idiotype antibody, it means that antibody will react here. For example, if donkey make anti-idiotype, that will react here. If donkey make anti-isotype, that will react here. If donkey make anti-allotype, that will react here. Is that right? Any question? Right? Class dismissed.